Hello, everyone. Welcome to Behind the Scenes at a COVID-19 Diagnostic Testing Center here at the Zuckerberg Biohub. We are really happy to meet you all and talk to you this morning. What we're going to do is start with a video for those of you who maybe haven't had a chance to see it on demand about the Diagnostic Center and how it all began. And then we'll come back and we'll chat with our panelists live.
This is the kind of pandemic that we all wish never would have happened. We knew we were facing a surge of cases that were coming our way. There's hotspots cropping up all around the state, and we needed more capacity and we needed it fast.
We kind of already knew that something was brewing, but there were a lot of challenges. You know, we weren't getting any reassurance from the CDC that we were allowed to do any testing.
On the 12th of March this year, the governor of California changed the regulations to allow some previously only research labs to do clinical diagnostic testing.
Giving you a sense of where we're going is to centralize our testing capacity, to partner with our counties, and to provide facilities where we can access substantially greater number of tests.
And within, I think, probably less than an hour of that executive order going through, Joe had gotten a call from the clinical lab at UCSF, and the decision had been made that we were going to ramp up testing. We turned our research lab, which was essentially just empty lab space at the time, into one of the nation's leading COVID-19 testing labs, capable of processing thousands of tests per day, and we did it all in 8 days.
Many of these departments of public health don't have good access to public health laboratories with large testing capacity. So in many cases, especially for the safety net hospitals, if we weren't doing this testing, the testing might not actually get done. So it turns out that the Biohub is the perfect place to build one of these expansion labs. Many of the problems and obstacles that we face required kind of novel solutions. We have a whole engineering team that works on robotics and electronics and fluidics. And so when we encountered a problem, we had engineers on staff ready to solve them.
We thought we were going to have about a week to set up the lab and get validated, and then I believe we were told we needed to be able to run our validation in 24 hours.
As research scientists, we're not set up to do this type of work and to understand how to navigate the FDA and the CDC and, you know, kind of all these systems that we just aren't used to.
And that, I think, was the moment I was like, I don't know if we're gonna be able to do it.
But we did it, and we did it in record time.
We all worked really late into the night, came back the next morning at like 5 a.m. ready to do the validation, and we passed.
Okay, so welcome to the CLIA Hub. Here, this is where everything starts. So we get phone calls throughout the day from 8 to 5 from all of the neighboring departments of health. They come and bring samples to us.
Hey, look at this, all organized and tests and everything.
Yeah.
Okay, all right. Thank you so much.
I got 6 of them in here for you.
Okay, great.
Just seeing the whole place just pivot like one of those transformer robots that you thought was a car and like all of a sudden is like a PCR testing crazy robot was really cool. Just it felt like all those relationships we had from all of these research collaborations just sort of transmuted, and that meant we all knew each other and could work together well. And with our partners at UCSF have also been incredible. Just the whole thing sort of stood up.
All of the electronics companies weren't really shipping because of the pandemic. So we ended up making a device out of scrap electronics that we had in the lab. We had a scrap small LCD screen, we had a small mini computer, and a barcode scanner.
This was made by our bioengineering team. We call it the Bartender, and it's essentially a little device for filling these tubes with Shield.
This device is called the Well Lit, and we use it to rearray samples from tubes to plates, and plates are what we use to do the RNA extraction.
This is called the Hamilton. It has all of the different patient samples, and we're going to move them from the individual tubes into a 96-well plate.
So what we have here is a Bravo liquid handler. What it basically is a machine with a robotic arm. The robotic arm will move up and down and around between the 9 different platforms, picking things up, moving things around, aspirating liquid, dispensing liquid.
So our computer engineering team, together with our data scientists here at the Biohub, spun up an entire laboratory information system in just a matter of days to literally do all of that tracking and reporting in a way that basically people said couldn't be done, but we did it in record time. So this entire process of booting up the CLIA hub at the Biohub has been a tight partnership with the Chan Zuckerberg Initiative. And many others have really helped us build up this laboratory information system that without which we couldn't have been able to accomplish this. And so I put the call out, all you graduate students and postdocs or molecular biologists, if you want to help and you want to get involved in the fight, you can volunteer for us.
When I first signed up on the volunteer form, there was at least 200 people within the first week who, just from our UCSF community pretty much, who had volunteered. It's really great to see that people are kind of stepping in and wanting to donate their time basically to work on something together.
So these are UCSF graduate students, postdocs, and even faculty members whose labs were shut down in March due to the shelter-in-place order. And similarly, they also wanted to contribute, right? And so they graciously volunteered, 40 hours a week or more in some cases. And these are highly trained scientists who were able to, quickly get up to speed in the laboratory environment. They've been absolutely stunning.
The volunteers start to come in, and they are really the ones that do all the hard work. They do the extractions, they start the qPCR, and they do at least 2 rounds of these on a day. And then starting at around noon, the samples from other counties start to come in. So then we organize those, we accession those, put stickers on those, and put them in plates, ready for the next shift to do more of these extractions and get results.
They come in every morning and they want to help. They're like, what can I do? And it doesn't matter if the task is, unboxing things, stickering things, or if it's something a little more complicated like an RNA extraction, they're always ready to help.
This effort that we've embarked in has really reinforced my desire to be in medicine and public health, really work in that overlap. And CLIA Hub is receiving samples from all over Bay Area. This experience has shown me how medicine can work so closely with public health and how they depend on each other.
So in addition to just testing individuals to see if they're positive for the virus or not, we also have a whole nother layer of analysis here at the Biohub. What we do is any of the samples that test positive, we actually take out of the freezer and we'll actually sequence the SARS virus that's in that sample. The reason why is it would provide a very rich portrait of all the viruses here and allow us to know when there have been new introductions, for example, from out of state, or whether there's continued transmission in different pockets, which then allow people to focus their efforts into these hotspots to interrupt these transmission chains.
Understanding the sequence of SARS-CoV-2 can help us to understand how it's transmitted and being circulating within California, and it can help our public health and healthcare workers take advantage of that information to design interventions or control measures.
So prior to COVID-19, one of the things the Biohub had been really deep into was working on a program we called IDC, together with the Chan Zuckerberg Initiative. This was a cloud compute platform that is designed to process pathogen data from sequencers. One of the principles behind IDC was to have as many countries on this system as possible, to set up like a virtual network of nodes that would be like sensors, so that as new emerging viruses coming from wherever they may come would be picked up and detected rapidly in almost any population. Providing these tools really helps democratize the situation and put sequencing in the hands of those who actually really need it, but who haven't had access to it up till now.
It feels really good to be contributing and helping people and be putting my skill set to use in a way that really makes a difference in the world.
It really is amazing to see that the system can work if people want it to work. If the folks in charge at all levels start pulling red tape out of the way, they start focusing on the common good, it's amazing what we can do in even a very short period of time.
There's going to be challenges in the future for whatever job or whatever kind of project I'm doing, but with the right people and the right kind of efforts, and I think we could accomplish almost anything.
I don't think there's anything more impactful or more important we could be doing right now but this one thing.
Great. Okay, so now it's time for the live Q&A, and we're going to go to our panelists. My name is Christina Tato, and I'm here with also my co-host Manu Venashat, who's going to help us out moderating the questions from the audience. And please feel free to type in. I think we can try to use, I guess the chat is fine. People have started to use the chat. Before though we start and get into this, it would be great if we could have our panelists introduce themselves and tell us a little bit about what their role was in setting up the CLIA Hub. And so just very briefly, we have Emily, who led this effort on board with us. We also have Christopher, Serena, and Robert, who all were scientists that were on board and helped set this up from the very beginning. So get your questions ready, and Emily, why don't you lead us off with an introduction? Oh, there you go.
There we go. Hi, I'm Emily Crawford. I'm a group leader at the Chan Zuckerberg Biohub, and my research focuses on developing different methods for detecting various infectious diseases. So when this all sort of started coming up back in March, it made sense for me to sort of convert what I was working on towards specifically COVID-19, and I ended up helping to build up this lab, and still at it.
Great, Serena.
Hey, how's everyone doing? My name is Serena Tamura. I'm a PhD candidate at UCSF, and I have been volunteering at the Biohub since March. So I was one of the volunteer graduate students who helped with the RNA extractions, moving all the samples around into different plate formats. I'm so happy to be here to chat with you all about the look behind the scenes of this diagnostic lab.
Great. Robbie?
Hey everyone, my name is Robbie. I am currently a UCSF PhD student, but before I joined the PhD program, I was on the bioengineering team at Chan Zuckerberg Biohub for about 3 years. And as Emily and others started forming this COVID testing team, they brought me on board so that I can help them build tools to solve whatever emergencies might come our way.
And finally, Christopher.
Hi everyone, my name's Chris. I'm an MD PhD student at UCSF. And I got on board because Emily and Vita reached out to me. I'd worked with them in the past, and I thought this was a really great opportunity to be able to contribute during the pandemic. I study Zika virus, so basically I was working on the last outbreak up until this one happened. So it's not my first time working with viruses and doing this kind of techniques. And so I helped a lot with running the actual tests themselves, as well as doing a lot of the back-end work, making sure that the samples that we were receiving were able to get connected to the patients who we swabbed to get the COVID tests for.
Great. All right. So we do have some questions coming in, so we can just go in the order I think that they've come in. So one of the audience members asked, are these tests accurate compared to your longer tests? So they're referring to the rapid tests that some people can get. And they're saying that they're a little confused about the different tests and what the difference is between the tests that we run at the Biohub versus these rapid tests.
I can answer that, Christina. So the test that we run here is using what we call Rt Pcr, and it's a test that with very, very high sensitivity detects the viral genome. So it's an RNA genome of the virus. And it's sort of considered the gold standard for testing of really any infectious disease. The viral genome sequence is unique to COVID-19, so it's relatively straightforward to develop a test that looks specifically for the COVID-19 genome and you know, doesn't detect anything else. There we go. Vita's going to show some pictures. Nice. The downside of RT-PCR testing, as you all probably know, is that it is time consuming and it requires you know, a laboratory. It's difficult to do it in a sort of point of care type of setting. So there are some newer tests that have just been coming out over the past several weeks that we like to refer to generally as antigen tests. And these are tests that instead of detecting the instead of detecting the RNA, the viral genome, they're actually detecting some of the proteins within the virus. So it's a little bit of different biochemistry, and these tests are much more rapid. This is what you think of when you have like a paper strip test similar to a pregnancy test, and you can see a band appearing. And the way that works is that you have essentially an antibody that reacts to one of the proteins. So the protein is acting as the antigen in this case, and you have a labeled antibody that will react to that and show up as a color band on your paper strip. As I said, these have just been starting to come out in the last several, maybe couple of months. They're a lot harder to develop than RT-PCR tests because when you look at the RNA genome of the virus, you're just reading a sequence, a series of letters, A, T, C, and G. And when you're trying to develop an antibody to a protein antigen, it's a lot more complicated because you have to deal with the three-dimensional structure of the protein. So it takes time for those to be developed. And in addition, generally speaking, in the world of infectious disease diagnostics, the antigen-based tests that detect proteins are less sensitive and less specific than PCR tests. So that means that you can have false negatives, particularly when the titer of the virus in the patient isn't high enough. So if there's just only a small amount of virus present, may not be picked up. And you can also have false positives. It's a less specific test. So there may occasionally come times when you detect a virus, but it's not there. Luckily, we've seen that the tests that have been coming online recently are pretty good. They're definitely not as good as PCR tests, but given the benefits of having a rapid and cheap test, we think that in terms of helping our community and helping us all sort of return to normal, that there are some really, really important uses of these rapid tests in combination with the PCR tests.
Great. And actually, while we're on the topic, another question came about how PCR actually detects COVID-19. So maybe we can give a very an illustration, or we can talk a little bit about how actually the test does work that we do at the Biohub. And we have maybe another slide that might help, but who wants to tackle that question?
Serena, I bet
you
got a good answer for that.
Yeah, I guess we can all kind of jump in and chime in together here. So the way I like to think about it is qPCR is a really good way to detect is the viral genome in this sample? So it starts with, you get your nasal swab, it goes into a tube, what happened, right? So what we do is we take the liquid that the swab is sitting inside of, and we say, is there COVID RNA in it? The way we do that is we extract out just the RNA from the sample, and then we use these probes that measure fluorescence and accumulate to see, is there any of that specific COVID RNA in that sample? And it uses what's called a thermocycler, which kind of heats the sample. It heats the sample to a certain degree, and then it amplifies and amplifies and amplifies to a point where we can actually detect that fluorescence. And so that's when we are able to say, hey, there's a viral RNA in here. That's a positive sample. We got to follow up. So I don't know if someone else wants to go through the CT values and the different and those kind of nitty gritties, but this is a really good, the screen share here is a really, really good example of kind of a qPCR trace. So I guess I'll try to go through it here. So on the x-axis, which is the horizontal line here, that's the number of cycles that, that the experiment is doing. That's the number of cycles of amplification. And so once it reaches this green vertical line, we can call that, what is that, the threshold for detection of the viral sample. If we see anything that crosses that threshold, that's when we know it's a positive sample. If nothing, nothing accumulates to that point, you're clear, no COVID. So qPCR is a really, really great way of measuring really, really specifically the amount of what RNA you want to look for.
That's great. And we had another question, what is RNA? And what's the difference between RNA and DNA?
RNA is a nucleic acid. It's very similar to DNA, and it's actually thought to be the original genetic material billions of years ago when life was first starting to form on Earth. It's thought that there were initially only organisms that had RNA genomes, and then somewhere along the very early evolution, some cell figured out that converting from RNA to DNA would have some benefits. DNA is a little bit more stable than RNA, but there remain several viruses that are relevant to us today that have RNA genomes, and there also are many viruses that have DNA genomes.
That's great. Okay. Another question that we have from the audience is, I noticed that the samples were brought in cardboard boxes, but after analysis, they were placed in a freezer. How long can a sample be out and not affect the results to detect the virus? Great question.
That is a great question and something that we all care a lot about. So the general idea is when you saw those tubes and all those cardboard boxes, those were all stable at room temperature. But after we do what's called the RNA extraction, when you have just the pure RNA, that's super unstable and needs to be kept, if you want it to store it, it needs to be frozen at -80 degrees Celsius at the very least. So depending on where you are in the experiment, the actual nucleic acid storage conditions changes.
Great. Also depends on what kind of media the sample is collected in. So you'll find that there are some tests run at some labs where the sample is less stable and really needs to be run within a number of hours or just a couple of days of receiving it. We actually use a media called DNA RNA Shield, which is a protective media that helps the RNA stay intact until we're ready to test it. But of course, we try to run the tests as quickly as we possibly can when the sample gets in, because we know that it's really a problem when there are long turnaround times and people don't get an answer to whether they're positive or not for a number of days, and then they've lost potentially the opportunity to be isolated, to get treatment, et cetera. So we try to be as fast as we can.
Great. So let's have a bioengineering question here. So saw some really cool kind of robotics going on in the background, and maybe Robbie, you can talk about one of those special robots and why we needed it and what it does.
Sure. So the robot that we have on the screen right here is what we call the Bartender. And its purpose is basically to fill all these empty tubes with a solution that protects the sample when it's collected. So normally we wouldn't need to build something like this. We could just buy it from someone else. Usually companies make these things. But back in March, we discovered that Italy was the main producer of these tubes for the whole world. So when Italy got hit pretty heavily by the coronavirus, they had the company that made these basically shut down. So all these facilities like UCSF, Stanford, everyone else, they couldn't actually buy any more of these tubes. So in order to help make sure that the whole state of California can continue testing, the bioengineering team made this dispensing robot so that we can make our own tubes inside the testing facility. And once we made these test tubes, then we distributed them out to the departments of public health throughout the state. Then there's also another machine called the WellLit. And what this machine, so during the transition period, even though like Italy was not able to produce more, a lot of facilities still had these tubes. They just couldn't buy more of them. So they were still testing with these original tubes. And in order for our facility to actually check to see if COVID was in the patient sample, what the facilities would have to do would be transfer the solution that from the normal collection tube into the buffer for our special, I can't describe this very well, but yeah, sorry, can someone help me?
No, that's great, Robbie. I can, this one is a little bit hard to understand but it's hard to understand maybe if you haven't worked in a lab, but if you have any experience working in a molecular biology lab, you'll know that we often spend a lot of our time figuring out how to do things in higher throughput by transitioning from single tubes, like you see there in panel B, to what we refer to as 96 well plates, which you can also see in this image here. Essentially, once you have 96 samples in a 96 well plate, you can do kind of one reaction with them all at once rather than having to do it 96 times. And biologists are really obsessed with the number 96. I'm not sure how that started, but we love 96 well plates. So what we need is a way to actually transfer the 96 different samples into the plate and keep track of which sample goes into which well, so that we don't lose that information later on and lose track of whose tests went where. So that's what this device is designed to do. And the bioengineering team at the Biohub, Robbie and a few others who use saw in the video, is just absolutely amazing the way they were able to put this all together, like literally over a weekend in early March. It helped us so much to be able to really keep track of what sample we were putting in what location. We really would have been so lost without something like this. It's really great to have that that creativity and resourcefulness come in. Same with the Bartender as well. Both of those are just, I'm so proud of those two instruments having come out of this project.
Great, thank you. So we did have some questions about how we know the test is positive and what is the CT value used to determine, or what do we use that for? Christopher, you want to field that one?
Yeah, absolutely. So I think, let's see if the question I saw correctly was, how do we know we're picking up COVID-19 and other coronaviruses? That's a really great question, because other coronaviruses actually cause the common cold. So it's really important to be able to differentiate between COVID-19 specifically and other similarly related viruses. The way this is done is that when we do these qPCR tests, we actually have very specific targets. So like was mentioned earlier, we're targeting the genome or the genetic information of the virus, which is an RNA molecule. So similar to DNA and RNA, there's a very specific set of letters that make up the genetic information of this virus. And so that's different enough from other viruses that we can specifically target one versus the other. To make sure that we're actually picking up COVID-19 and not other viruses, we will also test them against samples that we know don't have COVID-19 or may have another coronavirus, just to make sure there's no interaction and it's not accidentally picking up a different virus. That's a really great question. As far as like the CT values go, this is a little bit of like a technical question and kind of a, the cutoff itself can be kind of arbitrary, but if I remember correctly, we were using CT values of 30 or 35, which is pretty dilute. The higher the CT value goes, that means the less of the RNA that's actually there. It means you have to amplify the RNA more and more to actually detect it. And so I think each lab does it a little bit differently. I don't know if someone can clarify the exact number, but I don't think that's the important part. I think the important part to know is that the fewer amplifications required to detect the virus, it usually means the more virus is actually present in that sample.
That's great, perfect.
It's actually our current limit is we go up to 45 cycles. We initially, the first version of our test, I believe went up to 40, and we made an adjustment recently to it, so it's a bit more sensitive.
Perfect, thank you.
That's great, okay.
Yeah, there was another great question. Somebody was asking in the video, some of the staff are taking nasal swabs, some of them are doing something with people's forearms. Can you clarify what is happening and how does this correspond to the tests that are currently being described?
Yeah, I can go ahead and take this one as well. So here, let me just pull up the answer so I can read it exactly. Let's see, so in the video where it's showing the people taking the nasal swabs, what that's doing is actually trying to detect virus itself. So viruses and many other different pathogens like to infect specific parts of the body. As you guys know, COVID-19 is mostly found in like the lungs and the airways. It causes like a cough and problems breathing. And so the virus itself is actually replicating in your nasal passages and in your the parts of your body that you use to breathe, so like your lungs, your airway, that area. And so when you do the nasal swab, you're picking up the direct virus itself. The blood, or where they were doing the test of the arm, what they're doing there is actually drawing blood from patients. So whenever you're infected with a virus, or like if you get a vaccine, you generate an immune response which creates antibodies. These antibodies are molecules that help protect you against future infection. And so usually after you have an infection or a vaccine, these antibodies will stay in your blood for quite a long time. And so in that case, we're actually trying to pick up antibodies specific to the coronavirus, to COVID-19. And then this is another technicality, but COVID-19 is very rarely found in the blood itself. So when we're doing that, we can make sure that we're just finding the actual antibodies themselves.
That also just reminds me to mention that part of what's been so great about this experience is being able to work closely with UCSF clinical researchers doing community-based studies of COVID-19, and it's just been amazing how much effort there's been, especially in the Bay Area, but I'm sure this is true all around the country, to get testing out to the communities. And really have it be something that that is available to anyone who wants it or needs it. And in many cases, we collaborated along with clinical researchers who set up these tents around various neighborhoods in San Francisco or the East Bay. And people were able to come up and get a COVID-19 test to see whether they were currently infected, and then in some cases able to also get the blood test. And those two pieces of data that tell about current infections and tell about likely past infections, when put together, are really powerful in an epidemiological sense to understand the spread of the virus.
Great, thank you, Emily. Thank you, Christopher. Okay, so let's see. Manu, do you have a question queued up?
Yeah, there was one by E. Lin, and she was asking, when you are processing so many tests at one time, how do you ensure there are no errors, like assigning a positive result to the wrong patient? How common is such an error at your lab versus the commercially available point of care Rt Pcr tests like the Abbott Now? Maybe Emily?
Yeah, that's a great question, and definitely is something that keeps us all up at night, being just incredibly concerned about reducing errors to the absolute lowest rate possible. We have a lot of different ways that we have checks on what we do to make sure that no errors occur. So these are everything from checks at the sort of data entry level where samples come in and we need to check that everything is exactly correct with the labels and the barcodes. We have a number of places in our process where there are barcode readers that will help us to keep track of things so that we don't have, there doesn't have to be sort of human read. Oh, nice. This is an image about our data flow. We do have also, so you saw in the video that there, although we keep the samples separate sort of in these different wells of the 96 well plate, that there are times when the plate is open and exposed, and there is the small risk of cross-contamination from one well to another, and we're extremely concerned about that. We've done extensive testing on our assay as we were starting out at the beginning, and throughout this time, the past several months, we've continued to do this testing to understand how big that risk is, and to understand at what points we need to go back and repeat a test because there's a fear of the possibility that occurred. So we have a process for that, and that does happen where we go back and kind of double check. In terms of how accurate or how error prone or error free our test is compared to commercial ones, that's a really good question. That's a little bit hard to answer because I think every lab has its own sort of internal validation data, and we all have to meet certain standards set by the FDA to prove that we're able to detect a certain frequency of positives and negatives. So there are processes where, for example, you have to have a certain number of tests that are performed at your laboratory, and then the same exact samples are forwarded to another laboratory that already has a validated test and repeated there, so that the FDA knows that there's a concordance there. So beyond all testing labs that have either FDA approval or what's called LDT status, laboratory developed tests in a CLIA lab, all having met sort of basic standards set by federal and state government, it's difficult to answer that that question more specifically. But I will say that different types of tests have different, as I was mentioning earlier, rates of false positives and false negatives, and they also have different levels of sensitivity. So our test, we know, is extremely sensitive. We can detect 100 viral genome copies per milliliter. There are some other tests out there, including, for example, some of the antigen tests that I was mentioning earlier, that we know from the data are much less sensitive. So, for example, they might detect only something like 10,000 or 100,000 virus particles per mil. So it's important for clinicians and public health officials to know these parameters about any given test when they're considering the test results, both in a clinical setting and a sort of epidemiological setting.
Thank you, Emily. That was great. We also have a little video of how the RNA is extracted from samples, and I think Vida is going to pull that up. Serena, do you want to talk about the different steps that you'll see?
I'll try to. Let's see here.
Yeah.
I don't think I've watched this video, but I've lived it many times. Okay, so what are we doing here? We are all in PPE, super safe, and now everything's ethanol sprayed. And now, do you see all those barcodes? That's what Emily is talking about. It is really hard to lose a sample because literally everything is labeled, scanned, barcoded, and then it's backed up to what I assume is the cloud or something. I don't really see that part of it. And then you can see all these different decks on this layout here, and that robotic arm is working really, really hard. It's pulling up all those 96 samples and then mixing it with all these reagents. I believe this is the mag bead step. So it's mixing together with these magnetic beads that's used to extract out the nucleic acid, the RNA that we're looking at. So it's mixing, mixing, mixing. And let me tell you, this is a non-trivial step. It's really important that the sample is perfectly homogenized or perfectly mixed in. And you saw that now the 96-well plate is sitting on this black deck right there. That's super special. It's a magnet. And that's pulling out all those magnetic beads just so we can keep only the RNA. So it's a really, the idea is really simple. You have a sample and you only want a certain bit of it. And this is what you have to do to go through it. In my research lab, not at the Biohub, I do all of this alone by hand with one pipette. But that's what was awesome about working at the Biohub is there are robots that help you do this. So our throughput is just so much higher. There's so much less room for human error. And it's really, really reproducible. So this whole process, we were able to get this down to maybe like 2 hours with that, for an RNA extraction. So it's really robust. We're able to do multiple samples at once. And you can see here, it requires a lot of being super diligent and detail oriented because you see that 96-well plate is moving all around. I call it the dance floor. It's moving around the dance floor of the robot. But the orientation that you put that 96-well plate matters. So every time you're relying on this robot, like you still need to be there and be super tuned into what's going on. What stage is it? Oh, we're doing another magnetic extraction here. And then soon we'll wash off all the reagents and just have pure RNA that'll be loaded into a qPCR plate. But wow, this is a really fun video. I never saw this before I started working at the Biohub. This is really cool.
It's almost done. It's starting the qPCR now.
It's starting the qPCR. The layout is different, but I have to really tip my hat to all the engineers who programmed this robot. It's so specific down to the millimeter of where it's moving. It's truly amazing, and I feel really fortunate that I was able to get trained on this because this is not what grad students get trained on. We get trained on the low throughput, suffer alone with one pipette kind of method. So this was super cool. I really enjoyed doing these RNA extractions.
And now you're all going back to your labs and demanding that your PI get
Exactly. Actually, one thing I wanted to add was before I thought label makers used to be my favorite thing. Now it's barcode scanners. Gosh, that was awesome. It's almost impossible to lose a sample if everything's barcoded.
So that's great. And along this line of being able to do such high throughput, yet we're doing still individual tests, right? And so one of the attendees has asked, some countries conduct mass screening by pooling samples together. And so they wanted you to comment about whether what's the difference between the two strategies and what are the pros and cons between the two? Maybe Emily, you can do that one.
If anyone else wants to jump in. Yeah, so pooling, so essentially what that refers to is, think of that 96-well plate that you just saw on the robot there. What if instead of a single sample in each well of 96-well, we actually pooled together, 5 or 10 samples? So, instead of having like 1 milliliter of one sample, you might have 1/10 of a milliliter of 10 different samples. And then you would do the extraction and run the PCR and see whether you got a positive in any of those wells. Now, if you do get a positive, all you know is that one of those original 10 samples, at least one of those original 10 samples was positive. You won't know which one until you go back and deconvolute the pool, which means go back and test each one individually. So the advantage certainly is that you can extract 10 times more samples at once. There are a number of disadvantages. One is that when you do have a positive, you have to go back and deconvolute. So that adds additional time before the person who is going to receive that positive result will be notified. So that's not great. You also have a reduction in sensitivity. So imagine if only 1/10 of that volume is represented by each individual sample, you actually have to have 10 times more virus present in any given sample in order to detect it. So when I mentioned before, our limit of detection is 100 viral genome copies per milliliter. If you had 100 viral genome copies per milliliter, but your sample only represented 1/10 of that, then you would only have 10 viral genome copies in that sample. So that would be missed. We would miss that infection. And then the final issue with pooling that makes it a little bit tricky, a little bit complicated, is you have to stop and do the math and figure out at what point, at what level of positivity, at what positive rate in your population is it no longer useful to pool. Because imagine if 20% of your samples are going to come back positive and you're doing it in pools of 10, well, the majority of your pools are going to come back as positive and you're going to have to deconvolute every single one of them. And that's just creating more testing work for yourself. So there's a lot of, you can do the math and figure out exactly what the positivity rate is that makes sense to do that at. So there's a lot of good reason to try those strategies when you have a very, very low positive rate population. So you're doing something like asymptomatic surveillance work. That's not what we're doing here. We are doing a lot of testing for places that are experiencing outbreaks. So either nursing homes or other congregate living facilities, or just communities that have a really high rate. So we've never been in the realm that it would make sense to do pooling, but it's certainly a strategy worth considering in the appropriate cases.
Great. So another question actually refers back to the Bravo video that we just saw, and it says, does the robotic arm contaminate the plates? Wouldn't you have to replace each pipette tip every time a new plate comes in? Great question.
That is a great question. And yes, you do have to replace the tips. The tip changes are all built into the protocol, and it's really smart. We know when, at what stage the pipette tip is just touching reagent, for example, that's okay to go back to the same reagent to pipette again. But if it's touching the sample, only one and done. So that's all built in. We call them tip dumps or whatever, and those go straight into the trash, biohazard trash.
That's great. Okay. Let's see. A lot of great questions here. It's hard. We're going to try to get to everybody's, but we are coming down in the last 10 minutes. Thank you so much for all of the great questions that everyone's been putting up here. Let's see. Okay, we've had a few questions about things like, who do you report to once you have an answer, and what kind of regulatory bodies or others that you report to? And also, I'm going to add this in together. Another person asked about Hipaa compliance. So we have someone who's very knowledgeable about patient privacy and wanted to know if everything is compliant in that way. Emily, you're probably the best one to answer this.
Sure. Yes, I'll start with that final one. Yes, we are Hipaa compliant. It's something that we think about all the time. We're very, very conscious of it. And actually, I would say that's why we aren't able to do sort of a real live tour where we actually, show you the lab right now. There is testing going on in the lab right now, but if we were to bring you in there, there would be a lot of patient information that it would be difficult to keep away from the camera. So that's why we had this this very carefully produced video that we showed you. And the answer to the question about regulatory and who we report to, well, I guess those are two different questions actually. From a regulatory standpoint, I don't think I'll bore you with too many details, but this lab is physically located at the Chan Zuckerberg Biohub, and a lot of the staff that initially started in the equipment and reagents belong to the Biohub. But we operate as an extension of the UCSF Clinical Microbiology Lab. So we operate under their CLIA license. We operate under the direction of their lab director and their microbiology director. So all of the sort of regulatory frameworks that we need to follow are already embedded in the structure that we use there, and we definitely could not have done any of this without them. This really is their project, and we just happen to have the resources along the lines of personnel and equipment that we're able to do it. Yeah, there are both state and federal regulatory frameworks that need to be followed, which are very, very important. They relate to HIPAA as well as to other patient protection measures. We also do the majority of our testing here in conjunction with public health departments around the state of California. So that's who we are actually generally reporting the results to. There's an automated system that occurs through UCSF where the results go back to the ordering physician. And so in some cases that might be an individual physician from a clinic. In other cases it might be the director of the public health lab of a county. And those counties' public health labs are then responsible for everything that happens downstream of that. And that's of course, something that they already have systems in place for. So, it's not our job to deliver results or to, give any sort of specific advice. We've really been sort of slotted into this role as scientists, where we're contributing what we know how to do best, which is build these awesome machines and run PCR reactions. And we leave it to the experts in terms of clinicians and public health officials to do the rest.
Yeah, that's great. So we are running close to the end of the session, and we really would like to close out with this last question from Tracy, which is, and I think I would like to pose this question to each of you individually, and I'd like to have each of you answer this on your own. And so what Tracy asks is, she's curious to know, what is the one thing that was unexpected or that you learned during this whole project, or specifically while doing testing at SARS-CoV-2? So one thing that was unexpected that you learned, or one thing that really stands out to you. So how about Robbie, if you're ready to answer that, maybe we can start with you.
Sure. So the thing that I was most surprised about is, as someone who builds tools, I often spend a lot of time, like literally months to even a year, building a super complicated instrument that can do one thing. And when Emily brought me on board, there was such a huge pressure to build tools that can help solve the problems that we were facing now, that we were able to build these very unique instruments within a few days. And these tools that we built actually are not very complicated. And yet they're able to have such a huge impact on so many people in California. By the time I left, we distributed like 150,000 test kits to departments of public health around the state. And in order to build the machines that could accomplish that, we really only needed maybe 1 year of electronics experience and fluidics experience. So yeah, what's surprising to me was that you don't actually need to have a huge amount of skills to actually make an impact. You just need to know when to use those skills.
Well said.
Great. How about Serena? We'll go next.
Yeah, I think piggybacking off of what Robbie just said is, so I'm a molecular biologist by training, so I do qPCRs, RNA extractions in my own lab like every day. The difference is from this experience at the Biohub, each sample was a life. It was a human life. And that was so profound to me. Being able to use the training that I received to conduct these very regular assays, experiments that I do regularly and actually help the community in such a time-sensitive manner is something that, that really struck me in a way that I was not really expecting. As scientists, we are really detail-oriented. We work with, specific model organisms, but I'm not used to working with clinical samples. I'm not used to the idea that someone is waiting for my results to truly know if they have an infection or not. So it really heightened, upped my game, I think, just in terms of making sure that you're super aware that you're doing the experiment down, perfectly, that you're super organized. All those things matter so much. And I'm so thankful that I had this experience to use my training to actually do something other than just what I'm working on for my thesis. Another thing that I'll just mention is I worked with the Hamilton robot a lot, which is the one that takes the your sample within the tube and moves it into the 96 well plate. And I don't think I've thought about snot more in my entire life. I'll just put that out there and close with that.
That's great. Okay, Emily, how about you?
Yeah, man, I really appreciate both of those, all of those answers. I think what's still just what I really like to think back on about those early times is just how it sounds a little cliche, but how everyone came together and worked together and built this team to address this sort of crisis situation. It's a little bit hard to put our minds back into the place we were in early to mid-March. But it was scary. And there were, I think everyone had reason to be afraid and to not know, what the future was going to be like. So it was just amazing to have so many people sort of come out of the woodwork and want to help. And amazing to sort of see what we could all do when we really built a team and thought about how to make an impact, for no reason other than to, really help our own community in this scary time. One other example besides the amazing volunteers that came from UCSF is that we had a ton of companies and other labs from around the Bay Area donating equipment and reagents to us. So during those first few weeks, there was like this constant stream of, somebody like the security guard desk downstairs would call us and be like, oh, you guys have a package. And we'd be like, we don't even know what this is. And just some box and it'd be full of like pipette tips and, came from some company that, had heard of what we were doing and thought it would be able to help. And that was so nice to get a view into how everybody has something that they can contribute, whether it's realizing that some pipette tips you have in your stock room could have an impact and shipping them over, or coming over like Serena and Chris and other volunteers and just doing more extractions that weren't for your thesis but were for clinical testing. Yeah.
I agree. The community effort was phenomenal and inspiring, actually. And so, Christopher, you get the last word, and you get to share your insights with us.
Fantastic. It might not be quite as happy of a note, but I think something that really surprised me with the COVID testing was really seeing some of the health disparities between who was getting infected, especially in San Francisco. We worked pretty closely with the Latino Task Force for COVID-19 and did a large study where we sampled just everyone in the southern part of the Mission near 24th Street. And just seeing the positivity rate, or how many people who actually were infected with the virus in that population, many of which were still essential workers and still having to work in person, I thought was not necessarily surprising, but a little disheartening. And just seeing how this was really affecting a different group of people. And so that's a published study that you guys should definitely go check out. But just as a reminder, like, these are human lives that we're working with, these are patients, and knowing the positivity or knowing the COVID status of some of these folks is really important to help stop the spread, especially in some of these more vulnerable populations. But seeing also the connection between the Biohub and some of these community organized groups, I think was really incredible, really trying to focus on those populations where we are concerned and there's a much higher risk of more serious COVID disease.
Great, thank you. Very important comments. All right, so we are a little bit over time, but I want to thank all the attendees again for your interest and your questions and your engagement this afternoon with us on a Sunday afternoon. And again, thank you to the panelists for sharing some more of your time with us to explain about what exactly the Diagnostic Center did, and thanks to everyone at the Bay Area Science Fair for allowing us to share our stories. Thanks.
Thanks for
Bye, everyone.
Thank you.
Bye.